is even more than the first class, which we used to know. We have the high, the middle, and the low. But right now, we really don't know the middle. We really don't know the middle class, because we are gradually being taken out of that class. We are gradually moving away from that class. We are gradually becoming extinct, only identifying the very high class. And so why are we saying this? Food security. It's not just making food available. We are talking about affordability also. Does it cut across everyone, the rich, the poor, especially the farmers who are really engaged with the practice of farming? Noting very well the earlier presenters who has talked so much on this crop. We know that this crop is domiciled with our poor farmers, and we need to take good care of them. So we now begin to see how to engage deliberate and intentional efforts in driving research towards productivity, not just productivity, affordability and availability. And that is why today I said that this program is highly insightful. And by this, it will give direction towards this crop. And thank God we are talking about African yam bean. This crop, Spinostylis stenocarpa, is indigenous to us. We own it. And so it is possible to do a lot of things with whatever you know is your own. We don't need to go far away to source for the genetic material. It's right there on our doorstep. African nations, we own this crop. And we see it as a climber. It grows vigorously. Unlike cowpea, it produces, somebody said the yield per hectare ranges between 1 ton and 4 tons, which is quite interesting. So if we're able to put in concerted effort, put in intentions towards research, we are sure to improve on this yield value. And again, we also look at the flowering ability. It has rich, promising flowering nature, which we need to really tap into. And then the various nutritional value is enormous. Talking about the seed, the tuber, and of course the nodules. It produces enough nodules to really serve the soil. And the flowering ability. Somebody talked about if you need to go hybridization, you really need to understand the flowering nature of this crop. With the little effort we have made, we were able to identify 3 major colors for the petals. The pinkish, the purplish, and of course the whitish green. It is whitish green when not opened. And then when it is open, it turns purplish or pinkish. We really need to understand all of this as we engage the crop in hybridization. Then it is important because it serves for food, for man, and for animals. It is medicinal. And again here I say it is a secure crop for food security. And then quality in nutritional value. Some people have worked extensively on this crop with respect to proximate and time trying to look at. I want to talk on so widely exposed. This moving the research investment via this crop. And so having said that, research into it, it is not just a dual producing crop. It has 3 products: the seed, the tubers, and the nodules. Some produce tubers, you think they are tubers, but they are nodules. And then again, all of this accounts for from this crop. Then the Guinea savanna, the characteristic that is common to Guinea savanna. Some are herbaceous crops. Assertions of the 8 assertions, to discover 10 of them having 3 good uses: producing seeds, producing tubers, producing nodules. And from this, 1 to 40 for 48. All those ones, relation is very important in talking about, sold by TSS 10 to be 32. Others that presented yield performance, at least I saw 2 slides here that actually I was looking at. Interaction affects endogamy. It's very important to see how to work on this top seed code and ensure that it comes out of the agronomists, breeders, and food scientists, biochemists. The product that comes in, we need to look at. The third one was talking about there's so much noise. And then, so what do? Ecological test across locations, across states within Nigeria and outside Nigeria, so that we'll be able to really identify the crops that does what? Your attention. And this is what I was able to find out.
Let's put our hands together once more for Dr. Aremu for that very interesting presentation. And to round up this section on agronomy, we have presenting now Dr. Ekajegbe Becky, who will be speaking
other crop aspects of the work. Now, with regards to effects of nutrient changes, what we did here was to use nitrogen, phosphorus, and potassium-based fertilizer, specifically muriate of potash, in the levolution of feedstocks using the leaf. Now, we also noticed that the muriate of potash within 6 and 8 grams per plant stand produced enhanced flowering, and also that eventually enhanced the yield of the plant. But however, superphosphate and urea did not have any significant effect on African yam bean within the fertilizer levels adopted for the study. Now we took another step forward. We did some research on the effect of salinity. The reason was because if you observe the Niger Delta area, about 70,000 square kilometers or so, it's an underutilized area, particularly very high in saline conditions. And then a study by Oputa in 1981 actually indicated that in the northern part of the country, where some cowpea leguminous crops were produced, increased fertilization usually would increase saline conditions in the soil. So we thought whether if we varied the salinity conditions of the soil, we would be able to adapt the plants towards such conditions. But what we actually had to do was to mimic soil salinity using analog grade chloride. So unfortunately, were that we had significant grain yield reductions. In fact, as high as 48% reduction in grain yield when you compare with the control. But we had increased accumulation of sugars in both the seeds and the leaves at higher salinity control. Now we took another step further. We looked at the usefulness of soil conditioners in the production of African yam bean. In this particular case, we were particularly interested in oil-polluted soils. We polluted soils and then subjected them to different remediation mechanisms. And what we observed was, irrespective of the mechanism that was adopted, particularly if they were not enhanced by substrate amendments, the plants just didn't do well. The plant only did well when the soil was amended. So that goes to buttress the fact that in production of African yam bean, perhaps soil conditioners should be adopted. Now, in the aspect of preliminary assessment of possible genetic enhancement, we did a survey in 2012 on there were actually three variant color varieties color variants in the Midwestern region of Nigeria around 2011-2012. And what we did was collect the samples and then we planted. What we observed was that the black color variant had the highest yield compared to the other two colors, actually the brown and the light gray. Now the question raised was whether the coat, the color from the seed coat had any effect on the plant's performance. Unfortunately for funds, we couldn't continue in that research line. I'm throwing that open now. Now, we took another step further. We thought, usually when I teach mutagenesis, we teach students the use of conventional mutagenic agents like sodium azide, hydroxylamine hydrochloride and all that. And then we just thought, what if we pre-treat these seeds and subject them to stress conditions? Would it improve the performance of the plant? We did that, and incidentally, we came out with very interesting results. We pre-treated seeds of soybean, well, not African yam bean this time. And, so we observed that it enhanced in oil. Now, another year came, I think that was 2013 or 14, when this Boko Haram rage was so severe. We usually would get sodium azide from Jos, and then it was too difficult to get. And we kept wondering why. Some people were saying perhaps it was because they observed that some people were using it as feedstock for munitions, so it became too scarce. And then the ones we got too expensive, and I just asked my students, why not rack your heads? And then we stumbled on these aminoglycoside antibiotics, ampicillin, rifampicin, chloramphenicol, the aminoglycoside. So we said, let's try it. And then we pre-soaked the seeds overnight in aminoglycoside and antibiotics. And surprisingly, what we got was the plant's survival was also enhanced. Well, it just remained at that. It's physiological basis for stress tolerance. This is very important because you might come and tell me TVC 151, TVC 50, whichever variety you name is this, is that, is good, is that bad. If you don't understand how the plants would, perform under different environmental stress conditions, you may not really tell a better story. We have engaged over 10 of our postgraduate students in this work. I just decided to bring out some of the titles of what my PG students are doing in the lab right now. One of them is looking at the reactive oxygen species, oxidative damage, and antioxidant defense mechanisms of 15 selected African yam bean. Another is trying to look at heavy metal mobility and distribution in cowpea, possible threat to biomagnification. The reason is because we actually, in previous research, we discovered that the seeds of cowpea could accumulate heavy metals if sown in heavy metal contaminated soil. So we were really worried. What if farmers invariably plant seeds in minefield areas? I remember there was a situation in Jos, I think Jos, when also, and then some said perhaps because the farmer planted the seeds where there was, I think, a lead mine or something like that. So we just thought maybe we should also do that and see whether there was any distinction in the capacity for the plants to bioconcentrate metals in harvestable plant parts of economic significance. One of my MPhil students is looking at iron toxicity on cowpea genotypes. Now, the reason why we chose iron toxicity is because in Edo State, the initial study I did in 2012 was centered within the Midwestern region. The crop is actually appreciated. There is a common delicacy called iyehe. I eat it, and Incidentally, within the Edo State belt, the soil is ferruginous, very high in iron. And then we are just trying to look at the possibility for iron toxicity for, well, not AYB this time, cowpea. And finally, one of my PhD students is trying to investigate variations among 4 accessions each for African yam bean, wild legume, cowpea, and soybean. Thank you very much.
Let's put our hands together again for Dr. Beckley. Yeah, his presentation. With his presentation now, we rounded up the aspect of agronomy and the general aspect. Now we want to go to the next session on this symposium on African yam bean, and that's on genetics and plant breeding. And to chair that section, I will call on my colleague, Mr. Benjamin Falloy. Let's put our hands together for him.
Thank you. So on my list, I have Dr. Adesoye, the next presenter. So we'll be presenting on potentials of induced mutation in African yam bean. Please join me to welcome Dr. Adesoye.
Good day to everyone here seated. As you can see on the screen, my title has changed. Actually, the work on mutagenesis is still ongoing, and I felt that it's too preliminary to report. And this topic was born out of a conference we had early this year on the Joint World Cowpea Conference and the Grain Legume that took place in Zambia, and where it was reported that cowpea as an African or as a crop that is highly nutritious and proteinous is also becoming deficient in terms of its ability to tolerate drought in the face of increasing climate change. And so it was suggested that there will be need to identify some orphan legumes that are stress tolerant and that will be able to, that will help to increase food security in sub-Saharan Africa. And so I've looked through and I've discovered that there's little or no report on African yam beans resistance to drought, or any work that has identified drought tolerant any variety of African yam bean that is identified to be drought tolerant. And so we just decided to, immediately after that conference, go ahead to screen some varieties of African yam being collected across different ecological zones of Nigeria to see their tolerance to drought. Now, one thing about drought is that it's a very complex trait, and the results sometimes might depend on at what stage you are trying to evaluate for droughts. Following closely with some of the recent works on cowpea tolerance to drought, there have been some good reports on screening for drought tolerance at seedling stage in cowpea. Some have screened at a flowering stage. And knowing that African yam bean is also very specific in terms of its requirement for photo period, it will be very, very tricky. I mean, trying to screen for African yam bean, one, because, I mean, especially at the flowering stage, because most of the time, it takes a long time to maturity, and so we just decided to quickly screen some varieties at the seedling stage, and that is what I'm trying to report here. We decided to focus on 3 areas or 3 kinds of characters: morphological characters, physiological characters, and biochemical from NAGRA, National Center for Genetic Research and Biotechnology. And we try to compare with one of the varieties of cowpea that have been known to be strongly resistant or tolerant to drought. So just looking at the picture diagram here, the methodology was actually to grow for 3 weeks, 21 days, and then to subject, after 21 days of planting, to 2 weeks of, and also 3 weeks of water stress. So here we can see the African yam, the control plants looking still moderately okay in terms of the morphological evaluation. And though there have been reports that African yam might be drought tolerant, but I mean, there's not been any specific documentation as to the level of tolerance. And here, looking at the water stress evaluation based on morphological traits, we found TSS 96 and TSS 82 to be one of those showing strong tolerance because of the high number of leaves. Then, based on the vine length, we also found TSS 113 to also be showing some level of tolerance. Now, looking at this, we have the cowpea below, the last on the list. It appears that it's not is much, lower in terms of its tolerance to stress compared to these varieties of African yam that are listed. Now, let me just go straight to some of the few parameters. We looked at relative content, we looked at number of leaf roll, knowing that leaf roll is one of the symptoms or one of the evidence of a drought tolerance. That means it's able to reduce its leaf area and thereby avoid evapotranspiration. So when the leaf rolls, it shows that it's a kind of adaptation to drought tolerance. And we also looked at number of leaf bond. At the tip also, you find some kind of brownish coloration when the plant is under extreme stress. And we evaluated this, and well, for these particular parameters, we looking at most of them, although statistically were not very significantly different, but the first one, TSS 96, like I mentioned the other time, was significantly different from the others, and the highest value here was found in a TSS 112, yes, along with 147. Okay, no, we also have 157 here. That was 2.0, showing a very high number of leaf bond. Then let me look at the biochemical parameters. We looked at proline content, total chlorophyll, and some other biochemical, some other metabolites. And we also compared among their sessions, and we also found 156 here to have a very high value, then 112 to also have a very high value of proline, 745. Length and physiological characters like leaf roll and leaf bond were increased under water stress compared to the control. And relative water capacity also reduced under water stress. There is decrease in proline accumulation under water stress conditions for their sessions ahead, and there was also some changes observed in the total proline compared the stress and the fully watered plant. After 21 days of water stress, watering was resumed and all their sessions recovered. But after 28 days of water stress, TSS 96, TSS 147, and TSS 95 did not survive after watering was resumed. Why this other variety from Nagrab, NGA01-08096, could not withstand the 28-day water stress and eventually died. From this research, this variety from Nagrab, then TSS113, TSS140, were concluded to be drought tolerant, having the features of, showing features of drought tolerance with high proline content, reduced relative water capacity performance, and reduced chlorophyll compared to other accessions. Recommended as drought tolerant for further research in African yam. Thank you for listening.
Please, let's put our hands together for Dr. Adesoye. So the next presenter,
which also points to the fact that value addition is very, very important, which I think the lady she mentioned it already. So we all know about African yam bean, and my first contact with the crop was during my master's. My supervisor suggested the crop, and I thought that generated interest in me, that out of the, that the issues that we need to deal with is food insecurity. We're trying to find solutions to food insecurity. And I know that Adewale et al. In one of their publications mentioned that we cannot depend on just a few crop species and expect that we would have food security. So in order to make sure that food security is a reality, we need to tap into the benefits that we have in African yam bean and try to find solutions to the constraints that we have. I'm going to lay emphasis on work that has been done at my institute. We have done some germplasm collection, and those germplasm collected were characterized. The nutritive and anti-nutritive content of African yam bean is a major problem that should be tackled, and also some scientists in the food and nutrition department have also been able to find what is the nutritive and anti-nutritive content of African yam bean. We've worked on the economic evaluation of African yam bean under intercropping, because that is how it is usually planted. We do not have it so much under sole cropping. And African yam bean is also suitable, as we know, for supplements in ruminant feeding, and seed quality of African yam bean has also been worked on. For me, I have worked on 23 accessions of African yam bean, which I collected from IITA, trying to find out the early vegetative growth parameters and the diversity among those materials. But currently, what I am doing is genetic diversity studies using SSR markers. And I'm also working on some materials I collected. Of course, stage of development is plagued with one constraint or the other, and that is why, even though the core on which I think everything can stand on, we cannot do without all the other sciences. And that is the way I think African yam bean can be brought to prominence. Thank you for your attention.
Thank you, Ms. Adeola. The next presenter is Dr. Clement Afolabi, and he will be presenting on assessment of some African yam bean for resistance to pests and disease under natural infection. Please join me to welcome Dr. Afolabi.
Good morning, everyone. I brought good tidings from FUNAB, and I want to appreciate the GRC unit for giving me this opportunity, which is more or less a feedback on the accessions that we collected from the unit. Yeah, I'll be talking about assessment of some African yam bean accessions for resistance to pests and diseases. Yeah, a lot of people have talked about the background, but ours is an ongoing work. We have done a field work during the wet season of 2015, the dry season of 2015,
and we still have some that are ongoing on the field. So we are screening about 40 accessions for resistance to flora, flower bud, foliar, flower bud, sorry for the mistake there, and pod diseases. We are also screening some for insect infestation, the damage on the leaves, flower bud, and the pod. My brother, Dr. Ogawi, is also my twin brother here, has talked about insect infestation. I only just want to confirm something on what he has done. And we are also trying to look at the yield loss due to insect damage. This is a percentage frequency of fungi associated with AYB leaves. Like I said, it's an ongoing research. They are still associated. We need to do a confirmatory test, that's the pathogenicity, to determine which of these organisms are actually causing some of these foliar diseases. And we'll see some of them later. Also on AYB, these are some of the associated pathogens that you have there. On the flower bud, these are the associated pathogens, and we'll look at some of them now. For the insect damage, I want to confirm what my brother said. Strep diosteti is the major insect pest of AYB. Some of the floral diseases. We are seeing blight, different forms, which will have effect on our yield. We are also seeing leaf blight, who will have effect on our yields, different ones. Web blights, different diseases during the wet season and the dry season. We also have wilting occurring. We have mosaic, it's a viral symptom or viral symptom. On the pods, you can also see the sign. That whitish thing we are seeing is what we call the sign of the pathogen. So all this will lead to reduction in yield. These are some of the things that we are still working on. We want to determine which of the organisms have associated some of these with the initial figures have shown, or the graphs have shown. Still part of the pod diseases. On the buds, you can see the healthy one, and you can see the rotted flower buds. Actually, this caused a very great, because we planted under natural infestation. We didn't apply any chemical, whether insecticide, whether fungicide. We didn't apply anything like that. So and this has been a very great problem on the field. And this can also be attributed to insect infestation, because once insects attack, it gives room for pathogen to attack. You can see the flower bud. Now, some of the damages caused by insects. You can see perforation of the leaves, even the buds. The pods and the buds, you can see different damage, insects on it. So these are part of my group. They are postgraduate students, one working on the pathology or the mycology aspect, while the other guy is working on the aspect. Thanks for the...
Thank you, Dr. Clement. The next presenter is Dr. Masha A. Azeke, and he will be presenting on biotechnology as tool for improving the utilization of African yam bean. Please join me and welcome Dr. Azeke. Easy, go up.
Okay, back. Okay, there. Good morning, colleagues. This topic is a little bit of a misfit compared to this section we are in now. We have been looking at different methods of processing the yam bean seeds to improve their utilization. I'm a nutrition biochemist. We have looked at different colors of, we use colors to determine the seeds that we worked with. Now, I went to a village in Akuku, Edo local government called Makeke. It's in the northern part of Edo State. It's a village where there is high cultivation of these yam beans, where we interviewed most of the persons there, the farmers and the consumers, to find out what problems they are experiencing with the consumption of the yam bean seeds. Many of them complained of stomach cramps. Some, like another legume, talked about flatulence. A few talked about diarrhea if they take too much of it, while there are some that complained of dizziness. Then there's this normal problem of the long cooking time. It takes between 4 to 6 hours to get the seed soft. The first three problems have been attributed to the presence of the alpha-galactosides, what we call the raffinose family oligosaccharides. So what we did, the preliminary studies, we cooked the African yam beans for about 4 hours, and we observed after 4 hours of cooking, there was only about 17% reduction in the alpha-galactosides. But for other legumes like the cowpea, the faba bean, the lentils, or the bicoma beans, boiling for between 40 to 60 minutes resulted in between 40 to 100% reduction of the alpha-galactoside. That shows that for the African yam bean, traditional cooking is not very effective in reducing the alpha-galactoside. So we started to subject the African yam bean seeds to different unconventional processing methods. The main aim is to reduce these phytochemicals like the alpha-galactosides and the enzyme inhibitors and many others. And also, the processing method should be such that will not require the expenditure of a lot of cooking fuel. We tried different methods. The first one was fermentation using the tempeh fungus, Rhizopus oligosporus. We also fermented with Lactobacillus plantarum. We fermented with indigenous bacteria. And then on a different area, we try to see if we try to sprout the seeds to find out when the phytase enzyme will have its highest activity. Like somebody mentioned phytase enzyme a while ago, so that when these sprouted seeds are fed to birds, they can better utilize the phytate phosphorus. And then we're able to get something out of that. The next stage is using different drying methods to see how we can preserve the phytase activity for utilization. We just got a donation of the freeze dryer. That work is ongoing. But for the fermentation studies, I'm going to report only one of them here, which is the tempeh type fermentation. It's a meat-like legume food that is very popular in Southeast Asia, and its popularity is increasing in USA and part of Europe. It's mainly made from soybean. The raw beans, first of all, dehulled and soaked for some time. But for the African yam, we soaked for 24 hours. For soybean, they soak for a shorter period. And then boiled for only 30 minutes instead of the 4 to 6 hours. They're allowed to cool before it is inoculated with the spores of Rhizopus oligosporus. We observed that you can also take an old tempeh product used to inoculate a fresh sample without using the spores from the microorganisms. Allow 48 hours fermentation and you have your fresh tempeh. This is an example of a product you get when you ferment with the Rhizopus oligosporus. This is from the brown or the marble-like variety. But for the milky-colored variety, we didn't get what we were expecting. We attributed it to the fact that for this milky color, we found a relatively higher concentration of cyanogenic glycoside when compared to the black and the brown varieties. Then we decided to modify the method. Instead of using water for soaking and boiling, we just used 1% citric acid, and then we found that it worked very well for the milky colored or the white variety. Also, for the one that succeeded with the normal procedure, we found that we got a light-colored product that was sterile, both for the white and for these other varieties. In conclusion, we found that the black and the brown seeds are good for the traditional tempeh process. The white seed can be employed for the modified, which is the use of the 1% citric acid. You only need about 30 minutes of boiling, unlike the 4 to 6 hours, so it's less energy intensive. And all other results you can find in a book that will contain some book chapters and some other journal articles there. They are available for you to read if you're interested. So you can wake up now. And this is a product. After the yam bean fermentation, you can decide to fry and use as a meat replacement for vegetarians. Thank you very much.
Yeah, thank you, sir. I think after this presentation, we are waking up. So I'll be calling Dr. Ashiru Olayinka. Oh, sorry. Dr. Aina Ademola. Please, Dr. Johnson is not around, as I was told. So the next presenter after Ademola will be Dr. Ashiru Olayinka. Please get prepared. Please welcome to the podium, Dr. Aina.
Good morning. My name is Ademola Aino, and I'm going to be talking on genetic diversity studies using phenotype, SSR markers, and the next generation sequencing of African yam bean accessions. African yam bean is one of the most important tuberous legumes in tropical Africa, and it can grow up to a height of 3 meters depending on the height of the stakes. They are grown for their seeds in West Africa and for their tubers in East and Central Africa, although the seeds can also be consumed. It takes about 90 days for the African yam bean to flower and about 210 days for the pods to mature. The protein content of African yam bean seeds and tubers is between 15 to 29%. Amino acid values are higher than found in pigeon pea, cowpea, and bambara groundnut. Tubers of African yam bean contain twice more the protein found in sweet potatoes or Irish potatoes and 10 times more the amount found in cassava roots. The African yam bean is classified, has been said by previous speakers, it's classified under the neglected and underutilized species, and these have potential to curb food insecurity and hunger. The uses of African yam bean stems from food and feed to its sociocultural uses in Ghana and Africa. In Ghana, it is used to prepare a special meal during the puberty rites for the girls, adolescent girls. And among the Ekiti State people, it is usually used to prepare a special meal during the marriage festival, probably because it takes a longer time to get done, and maybe that signifies longevity in marriage. I don't know. It's also, as stated by, I think, Dr. Bimbola, it's used for curing of headache and acute drunkenness, although I don't think that's a panacea for being drunk. And then the African yam bean has potentials of being used as a botanical. The presence of the lectin compound, which confers on it a little amount of resistance to major pests of cowpea legumes, such as the Clavigula, Tomanticicolis, Maruca, Testulalis, and the rest. So I think it has the potential to be used as a botanical. Justification for my study, there are limited genomic tools available for the diversity study of orphan crops. Morphological plant descriptors could be inconclusive and misleading, and SSR cowpea markers have been said to be markers of choice for legumes without a reference genome. And also the GBS, which is a novel and high throughput sequencing method, gives room for comparative analysis across samples of African yam bean without a reference genome. The objectives of my study is to investigate the morphological variation present in African yam bean accessions, to employ SSR markers and next generation sequencing method in understanding the level of genomic diversity present in these accessions of African, the levels of genetic presence in African yam bean. For the basis of studying 2-3 experiments and then use SSR markers, the accessions have been grown in this greenhouse. Leaf samples were collected as usual, and DNA was extracted following the modified De La Porta method. DNA quality and quantity were checked, and 20 SSR markers were used for polymerase chain reaction, and PCR products were checked on the agarose gel electrophoresis. And the fragment analysis, for the fragment analysis, I'm going to be colloading 4 different markers, fluorescently labeled markers, and these are going to be denatured after adding the fragment analysis cocktail, that's the leaves and the dye. It will be denatured for about 5 minutes and placed in ice and then sent to the genetic analyzer. After the analysis, the peak analysis sizes are going to be scored using the gene marker, and the polymorphic information content is also going to be calculated using the power marker. The second experiment is on morphological characterization of African yam bean accessions. 50 accessions were planted last year in IIT Ibadan here, and characterization was done using African yam bean descriptor list by Adewale and Dumet. Also, multilocational evaluation of 25 selected African yam bean accessions are ongoing, present in Ikene and probably Ubiaja next year. In my subsequent slides, I'm going to explain on how I selected those 25 of the accessions from the 50 African yam bean accessions earlier studied. For experiment 3, it has to do with genotyping by sequencing. The GBS libraries are going to be performed in an Porter's lab in the University of California, Davis, following the method by Eshe. Libraries, the genomic DNAs are going to be barcoded and sequenced on a single lane ISEC Illumina, and a unique version is going to be used for SNP calling. Sequencing will be done at the University of California, Davis Genome Center, and analysis at the bioinformatics core. Preliminary results from the African yam bean tubers. On my left-hand side here, you can see the variation in the African yam bean tubers sizes and shapes. TSS 30B is giving off it look like a spindle shape. TSS 370 is more of round or oval. And pardon my picture, the TSS 209 should be irregular in shape then. On my right-hand side is just a picture of mature African yam bean pods. Yes, this result here is showing some of the markers used in this study, SSR markers, and how they were able to amplify genomic DNA of African yam bean. This is just to show you how, if you look closely, you see that there's a variation, there's polymorphism, and you can see the variation in the allele, in the band sizes. Here you can see this. This is quite higher than is found normal higher. So the SSR cowpea markers are shown polymorphism in African yam bean study. Selected the 25 value, and the have accessions that performed very well across all traits that are of relevance to the breeder, and below you have those that didn't perform that very much. You can look here on top. This is the best performed accession, and this is TSS. This is TSS 59B. Also ongoing is I have some other experiment going on by the side, which is cooking experiment to determine how long it takes for these particular accessions to get cooked. I'm employing 3 methods in doing that. The first is I've used the electric stove cooker to do that. And also, I soaked, the next was to soak and to cook. I soaked the seeds overnight for 16 hours, and then we boiled to see how long it takes to cook. Also, the firewood experiment is also going to be carried out, since this is what most of the indigenous people use in cooking of African yam bean accessions. Coming up in this study will also be the nodulation studies and nutritional profiling of African yam bean accessions. I'd like to thank my supervisors, Dr. Abati and Dr. Elori. I'd like to thank Mr. Oyatomi, Susie, Bianca, the entire staff of JRC. I'd also like to thank the sponsors of this program and paid through the grant to the University of California, Davis. Thank you.
Yeah. Please, I want to ask one question. Who is doing the cooking? That's great. And from your presentation, you are using firewood later on. Go ahead. Please, before the next presenter, I want us to rise up. Please, let's be on our feet. Seems it's getting cold. Want to do some exercise? Just stretch yourself. So when I say AYB, you say yes. Let's say yes to AYB. AYB? Yes. AYB? Yes. And the last time, AYB? Yes. Please, let's be on our seats. Thank you. So next presenter on my list is Dr. Olayin.
Ashu Olayinka is my name. The title of my presentation is Identification of Quantitative Trait Loci for Drought Tolerance in African Yam Beans Phenostylus stenocarpa. The nutritional benefits of African yam beans cannot be overemphasized. We have seen that in the course of the presentation so far. I will go straight to statement of problem. Worldwide, occurrence of drought has become endemic due to climate change that has ultimately altered planting season according to precipitation pattern. When drought stress coincides with irreversible reproductive processes, drastic reduction in grain yield usually occurs. Drought has been found to be the most recalcitrant factor affecting breeders, and as such has immense impact on food security for the people. Drought stress is a major cause of grain yield reduction crops. During seedling establishments and other grain filling stages, drought can cause complete crop failure or varying amount of biomass reduction and grain yield. Under monocrop production, just like Dr. Oga has said, grain yield, production of African yam bean has been grossly reduced due to grain yield under monocrop production. The aim of my research is to screen selected accession of African yam beans, identify two extreme qualities associated with drought tolerance, make crosses, identify quantitative trait loci, and also map these quantitative trait loci. Objective of the research is to identify African yam, two genotypes that shows highest and the lowest extremes as parental lines. We also want to develop an African yam beans anchored map anchored by SSR with reference to gene and genome sequences of cowpea and soybeans. We want to identify and characterize this quantitative trait loci linked with drought tolerance. We also want to establish baseline data on genomic resources, especially as it regards to express sequence tags and bacterial artificial chromosome, which are BACs. The methodology will also be divided into the molecular aspect and the breeding aspect. The breeding aspect will involve collection of accessions. We have collected some from the genetic resources unit here. We also intend to collect from the wild, from markets and local farms. After which we will screen for drought tolerance in these accessions using the wooden box method as described by Singh in 1999 and also used by Ewansiga and Singh in 2006. The accessions with the performance range, both for the extremes, will be selected for the final screening. Data on number of days taken to observe the first land death and 50% and 100% plant death will be recorded. We intend to develop a mapping population through a single seed descent that will raise to F8 population, which will be our recombinant inbred lines. We will phenotype these recombinant inbred lines, also using the wooden box method as used by Singh, as I mentioned earlier. SSR markers will be employed to screen the parent and both the recombinant inbred lines. And MapMaker software will be employed to perform linkage analysis and also do our genotyping. To MapMaker QTL to identify statistical analysis using GeneStat. The genetic basis of drought tolerance, developmental regulated, is vital for efforts to modify the impact of water supply on African yam beans. Thank you.
Thank you, Mr. Ashiru. The next presenter is Susie Shita, also known as Oyatomi Susie. She'll be talking on amplification of African yam bean genomic DNA using cowpea SSR markers. Please welcome Susie on board.
Thank you. Good morning, everyone. Okay. Okay. Yeah, my name is Shita Susan, and my topic is transferability of cowpea SSR markers in African yam bean. I'll do a little introduction about African yam bean. We've heard a lot so much. One thing about African yam bean is that it has the potential to boost food security in Africa due to its considerable nutritional qualities. But we all know that African yam bean has some numerous challenges. And one of the challenges is genomic tools. We regard other challenges. We wanted to do a genetic diversity study, and we know some markers have been used, like the AFLP, the RAPID have been used, but SSR markers have not been used for African yam bean. And to develop SSR markers, the genome of that crop has to be available. The genome, AYB genome has not been sequenced, so there's no way you could develop a marker for SSR markers for AYB. So we thought, what can we do to be able to make available SSR markers for AYB? We now decided to do a transferability study. Yeah, we did a transferability study between African yam bean and cowpea, because cowpea and African yam bean belong to the same family. And research has shown that in the family Fabaceae, which African yam bean and cowpea belongs to, there's a conserved region of SSR within that region. So there's a possibility of transferring one marker to another. So the molecular work we did was in two phases. The first phase was that we worked on 67 accessions on African yam bean. We did the DNA extraction, then we need to do the polymerase chain reaction. How do we go about this? There's no any protocol on ground. We had to do a lot of optimization. We decided to use 36 cowpea SSR markers, and our markers were of different categories. Some were the EST, some were the unit gene, and some were just the normal genomic SSR. So we had to do a lot of optimization. After the optimization, 13 of these out 36 primers were able to amplify the genome of African yam bean. So we went further on to do the PCR, And for our scoring, we did the fragment analysis. After that, yes, these markers are transferable. Let's use them to do a genetic diversity within the 67 assertions. Then in the second phase, these markers that were transferable, the 13 markers, we decided to increase our number of assertions. We say, okay, let's validate these assertions on 180. Let's validate the markers on 180 assertions, which we did. Then we decided to do another optimization again, and we increased the number of our markers. So from 13 markers now, we now have 16 markers which are working for African yam bean. So this is just picture of some of my gel pictures. The pointer. The pointer, please. Okay. So this gel shows, this is the ladder, and we have this primer VM31. It's a cowpea primer. If you see, during our optimization, we have to optimize with cowpea, and we have to optimize with the African yam bean. These two regions we are seeing here, they are cowpea, while these ones are African yam bean. So we can see that the amplification, the region that this amplification occurs is different in cowpea. Cowpea sizes seems to be higher up than African yam bean. This is another of our gel picture again that is showing this marker in VM37. Here only the cowpea just amplified. So there was marker. This VM31 amplified everything, but if you can see the last band which belongs to the cowpea, the bands are bigger up. I'm sure by the time you run them further, they are going to separate into 2 or 3 sizes. But for the cowpea, they are smaller than that. So there's a difference in the region that was amplified. So this is the diversity in dendrogram we're able to come up with. We got 2 major clusters based on our 67 assertions that we use. So in our second phase of the work too, this is an additional marker that we have, which is VUGM92. This is a unigene marker. Then this shows us our fragment peak on the ABI analyzer. This is the dendrogram we have. This work, we've written the manuscript, but it has not been published yet. But the previous one I showed, we already have a publication from that work. So the conclusion is that the level of diversity revealed in our study shows that the AYB collections can be exploited for further breeding program. Then even the percentage transferability we recorded by cowpea indicate their usefulness in AYB genetic improvement. Then based on the result we've gotten, there can be a possibility of doing a comparative study between AYB and cowpea. I want to acknowledge the following people. Most of the work was my MSc work. My supervisor, Dr. Albertine and Dr. Adesoye. I want to appreciate Mr. Oyatomi, all the GLC staff, and the Bioscience Center. Thank you all.
Thank you, Susan, for the good presentation. The next presenter on the list is Dr. Kupola Jacob. We'll be presenting on genetic intraspecific variability and cytology of African yam bean. Please welcome Dr. Jacob on board.
Good morning to everyone. First of all, I would like to appreciate Genetic Resources Unit or Center of IITA for giving me this opportunity, and at the same time, those that have contributed immensely to my academic progress. I remember in 2005, my supervisor then, Professor Adebute, gave me a note to GRC. Then the Dr. Falakasmi or whatever was the head then. And when I met her, we wanted to work on miscellaneous legume. And she said, have you seen something like African yam bean? And I remember my supervisor told me, when you get there, use orientation very well. I will do that. She asked me so many questions. I was somehow blank because she said that. I think this meeting is about what we have done so far, what we need to do, and to chart a research need for the future of African yam bean. A lot has been said about the diversity of African yam bean. I will not dwell much on that. We've carried out studies on the morphological or phenotypic studies of African yam bean, then rapid intraspecific variability, and at the same time, chromosome and pollen studies of African yam bean. For the phenotypic intraspecific variability study, we used 25 accessions collected from a GRU, and for rapid, we just selected 10 glaring different accessions, and we subject them to rapid characterization. Then the cytological studies of African yam bean. These are some of, these pictures show us some variability, the presence and intensity of pigmentation on the African yam bean, which we can also exploit. This shows spot formation on African yam bean and some other variations on the yam bean. One thing I noticed about the flower of African yam bean, though it clastogamous, but then we did some reproductive studies too, because our work then was the morphology, the cytology, and reproductive biology of African yam bean. We did something on the reproductive aspect, and we discovered that after 3 or 4 days, the flower bud do abort. Is there a way we can do that? Maybe if we can apply the kind of a tissue culture technique to circumvent that. Then the final, again, these are the primers that we use for the rapid characterization. And one thing about these primers is that they have not been used before for African yam bean, though adopted from the one that have been used for cowpea. And they also gave us a high kind of variability among the accessions that we use. This is cluster analysis based on the rapid profiling. We have 3 groups here, and most of them were not grouped along eco-geographical areas of collection. They were not grouped along that line. I think they were based on their genetic characters. Then the cytology, which I would like us to please take note here. Our study revealed the diploid status of African yam bean, which is 2n is equal to 22, and n is equal to 11. But among all the accessions, there's a kind of disparity, particularly with respect to TSS3, in which case chromosome count of 2n is equal to 18 were recorded in some cells of TSS3. And I think Dr. Adesoye from you also recorded something like a chromosome number of 22 to 24 or something like that. I think you recorded something like that. I think this disparity or differences, we can exploit it too in the areas of genetic diversity of African yam bean. Then the 2n is equal to 18 needs further verification and confirmation, as the count may be due to some error or whatever, or even segregational error. These are some of the results from our cytological studies: chromosome number of 22 and chromosome number of 18 were recorded. But majority of the accession were 2n is equal to 22, which is somehow reported mostly for Fabiaceae family. Most members of, if you look at Figna ubiculata, it's also 2n is equal to 22. And I think African yam is very, very close to Figna unguiculata. Then the pollen studies. This is in the area of reproductive biology. All the pollens that we work on, we discovered that they were all fertile. They were all fertile, and that is very good for a yield of African yam bean. The more fertile the pollen, I think the better the yield. So the pollen size range between this, let me quickly jump. This is our result. We can see some collapsed pollen grain shows a kind of sterility in some of them. But if you look at it, majority of them are somehow fertile. And the other picture also shows us the pollen structure and shape of AYB. Finally, because we want to chart a new course for our African yam bean, what are the implications of our result? Now, if you look at most of the genetic diversity reports so far, it has revealed a kind of high intraspecific variability, which is very, very important for genetic improvement. Then, like I said, they need to exploit this disparity in chromosome count. And the future research needs, since a great success has been recorded for cowpea, I think we can also record successes on the African yam bean if we apply some of those techniques that have been applied to cowpea, like the case of a tissue culture. Is it possible to use tissue culture technique, particularly in the area of embryo rescue? Then the manipulation of seed coat, fat tissue culture, the molecular techniques. And I'm happy that some other people are already working on the application of SSR marker to further work on the diversity or maybe to predict heterosis. Thank you so much.
Thank you, Dr. Jacob. We appreciate the presentation. And at this juncture, I want to assure you that we are on the right course. And besides, I'm optimistic that this gathering will yield a positive result. You may be wondering why I said this. We have an eminent personality in our midst, and I want to use this medium to recognize his presence. Is Professor Adegbite, Ondo State University of Science and Technology, is the VC, Ekitipupa. Please join me and welcome Professor Adegbite as he's presenting on African yam bean titled Morphocytogenetic Diversity Analysis, a Path to AYB Improvement. You are welcome, sir.
Thank you very much. May I greet us good after Jacob. So good morning, everybody. And okay. I want to welcome myself, firstly, to my home unit in IITA. Home unit in the sense that IITA used to be my first place of work after leaving the university. I work here as a research associate of Genetic Resources Unit, which is now Genetic Resources Center. I also want to thank the management of IITA and the GRU Center for the warm reception accorded me when I visited early this year. I was part and parcel of the people that characterized the cowpea germplasm in GRU and the miscellaneous legumes of a GRU, including rice bean, mung bean, and the rest. And since I left IITA, I've been placing my students on miscellaneous legumes, one after the other. So this gathering will give me opportunity to showcase some of the work we've done on African yam bean. Well, my presentation will cover 3 areas: morphological studies that reveal the morphological diversity of AYB, the chromosome and pollen studies that reaffirm the previously reported chromosome numbers and the newly reported one, and the future research needs of AYB. Well, what has been said about the introduction of AYB, we may not need to waste more time, but the imminent constraints on the cultivation and utilization of the crop needs to be noted. Long cooking habits, the laborious and time-consuming cultural practices, staking is mandatory for African yam bean due to the indeterminate nature of the growth habit. And it becomes so laborious for farmers who wants to have something, good yield from it. Fungal attack also, as reported by some of the presenters here, especially when the humidity is very high. Also, particularly the one caused by flower thrips and maruca is also a major constraint. Please, you permit me to use more than 5 minutes. So I also want to ask Dr. Oga, or Professor Oga, to let us know why African yam bean that contains the chemical that confers resistance on cowpea, and it cannot confer that resistance on itself. So there is need to look into that. I think it contains some lectin. So what we have done, we have worked on 105 assertions in all for intraspecific variability of the assertions. All the assertions we have obtained from BRU of IITA. 25 of them were worked upon by Jacob Popola and 80 by Adewale, which by God's grace I supervised the two of them. And also chromosome and pollen studies. From here, we can see some level of variability in terms of pigmentation on the stem. It varies from light purple to deep, no pigmentation to light pigmentation and deep pigmentation, as shown in D, A, and B. Also, pigmentation variability in flower was also observed. Also on flower pods, majority of them of the assertions have straight pods, but some have curved pods, as shown on the fourth assertion there. This shows variability in seed size and color of the AYB assertions studied. Variability was also observed on the eye color pattern of the species. You can see the one in here having tan color and black eye pattern color in the one below it. Then the variability in the color, seed coat color, and then tuber. The other time, somebody showed a tuber collected from a session from Congo Republic. So variability also exists in tuber production. This dendrogram shows a grouping of the assertions, 80 assertions studied by Adewale, and they were grouped into 4 groups. This is majorly based on floral characters or reproductive characters. For chromosome and pollen studies, majority of the assertions show 2n equals 22 as the somatic chromosome number, and all of them show regular meiotic process, which resulted in production of fertile pollen grains. However, in one assertion, TS3, chromosome count of 2n equals 18 was recorded in some cells. that particular accession, 22 was recorded, but some cells have 2n equals 18. And this became worrisome. I didn't want to believe that it's going to be a new chromosome number until it is verified and confirmed, because so many things could be responsible for this lower count. Some of them could be overlapping of the chromosomes. In the process of preparing slides for these materials, we need to press the cover slip to reduce the intensity of the stain. In the process of pressing, some of the chromosomes can get escaped from the cytoplasm. But that could also may not rule out the possibility of segregational processes that can lead to reduction in chromosome number. So there is need to verify this. For pollen, we discovered variability in pollen size ranging between 53.39 in TSS23 and 95, no, fertility, sorry, and 95.3 in TSS119. Pollen size also ranged between 66.15 to and 82.75. The pollen grains were tricorporate with 3 germ pores, fenestrate with scabrate ezine. These are some of the chromosomes. This A shows chromosome count of 2n equals 22. So be it for this also, the affections that have those pictures are listed below. This is a particular line, TS3, that shows a count of 2n equals 18 for this cell. This is pro-metaphase or prophase of one of the affections that's also showing 2n equals 22. These are meiotic slides, showing regular meiotic process in the varieties, I mean affections. This is n equals 11, n equals 11, and n equals 11. But in this particular one, a big chromosome was cited in that particular line. This is the early anaphase 1 of TSS3 that showed the count of 2n equals 18. And what is observed here, while some of the bivalents are already separating, as observed here, some are separated, some are fully separated, some are separating, some of them still remain intact, like this, not yet separated. This late disjunction or non-disjunction of these chromosomes that are involved in the bivalence can lead to chromosome number change, especially if the chromosomes that are the 2 chromosomes in the bivalent did not separate and go through anaphase. That means the bivalent will go to one pole, whereas the other pole will be short of a chromosome. So one pole will get 1 chromosome, the other one will be deficient with 1. If that gamete happens to be fertile and is used in fertilizing a pollen mother cell, that can bring about change in chromosome number as experienced in this. So what could have happened if that 2n equals 18 was naturally, if it naturally occurred, it's possible that there were cases of non-disjunction that involve 2 bivalents. Therefore, 2 chromosomes will be short in one pole, and there will be gain in the other pole. These are fertility tests done on the chromosomes, pollen grains of some of the accessions. pollen grains that are not stained are sterile pollen grains, like these ones, and some with collapsed margin. So there was chromosome size variation was observed in majority of the accessions. And these are the chromosome types found in the AYB accessions, which is common to all. So from this presentation, which is a blend of the work of Popola and Adewale, the following inferences can be made. And future research needs on the AYB put forward. Intraspecific variability in AYB is evident from what we have seen and what others have presented in morphological and sociological characters. Implicated genetic diversity, as reported here, is an assurance for possibility of genetic improvement. There is need for research on the following areas: amelioration of the constraints on cultivation and utilization of AYB. If the cooking time can be reduced, if the breeders can work on the growth habits of this crop so that the level of indeterminacy of the plant can be reduced, maybe from indeterminate to determinate, and then a throughput molecular diversity studies on AYB, and I'm also happy that some people are already working on that in IT and other places. Harmonization of phenotypic and genomic data for proper matching of the AYB assertions. That is also necessary, also pointed out by some of our presenters here, to match genotype with phenotype so that we can know this particular assertion has this particular trait, so that when we are looking for those traits, we know which assertion to go for. So now that this forum is coming up, I'm happy to be here, and I want to assure that I've thrown this AYB work behind me for some time, but I'm going to probe further into this 2n equals 18 count, and also the range as reported by Adesoye between 18 and 24. We need to properly work on that so that we can come up with a very correct karyotype for African yam bean. Registration of the peculiar botanical characters of AIB with respect to different assertions also necessary. I have another, I want to add another area that we need to work up to promote utilization of cowpea. There is need for serious utilization campaign for AYB, as it was done for soybean in ITA in the 1990s. I remember there was a policy then not to use coffee or tea during our refreshment or break time. Instead, soy milk was replaced with all these things. So if that can be done, even if you are starting from ITA, to make this AYB to be more acceptable to the people as it was done for soybean. Thank you very much.
Thank you, sir. We are happy to see you around us. The last presenter this phase is Omena, B.O. I wondered when, before he got his PhD, he was known as Prof. Now that he got his PhD, he came down to Doctor. So I want to welcome Dr. Omena Bernard on board, and his title is, his presentation is titled genetic diversity and in vitro studies in African yam bean.
I have the privilege of saying for the first time, good afternoon to everyone. For me, this is a homecoming because I began my career here at the GROC, where I worked for 6 years, but under in vitro gene bank. A lot has been said about African yam bean. What I'm going to talk about now is majorly from my PhD work, which was sponsored by the International Foundation of Science, Sweden. Most land ranges of African yam bean remain uncharacterized and are still in the hands of resource-poor farmers. Very few of them have been conserved in seed banks. So we need to carry out more exploration and assess the genetic diversity within these land races in order to enable us to improve on the breeding of African yam bean. So this will be my outline. This will be my outline for my presentation of what I've worked on African yam bean for the last 6 years. Now, 40 accessions of African yam bean were used for this study. 27 were obtained from the GROC IIT, and 13 accessions were obtained from Professor S. R. Akonde of blessed memory from the Institute of Agricultural Research and Training in More Plantation. Now, interspecific variability of 40 accessions were capped using 48 agromorphological traits. From that experiment, I'll just give a brief summary. We found out that we had 4 different seed shapes, and out of with 82.5%. Superization, as has been said, occurred in some accessions, not all accession experiments. 17 of them produced tubers. 12 out of the IR produced tubers, while 5 from the GROC. Now, superization is generally under genetic control and could also be affected by the environment. In other tuberous legumes like the wheat bean, and we show that short day length, day and night temperature of 25 degrees. We found that the sunshine hour was 2011-2012 at the field at IRT. Less in 2012, and we had more in 2011. Rainfall was less, but we divided for photo period and temperature. It could also be possible that short day period also affects polarization in African yam bean. 1 and TSS 15 evaluated. Tester color variegation. And also by the factorial coordinate analysis. While those from IIT, those from the southeast Nigeria, we all grouped together as well. This is a molecular tool for the assessment of genetic. I want to go further to see if we could develop SNP markers. What we did was to isolate that showed homology. After diluting and purifying the bands, we tried to sequence, but we later found out that the quality of the DNA was not good enough, so we stopped that research. I also studied the mechanism of pollination in African yam bean using 3 pairs of accessions from RITA and also from the accessions from Institute of Agricultural Research and Training, More Plantation, using the method of Daphne 1992 under field conditions. We allowed the plants, we covered the plant completely, as you see here, before flowering began. Some sets of plants were left to open pollinate. White crosses were made within accessions and between accessions. At the end of the day, we obtained a self-in and out-crossing rate of 91.38% and 8.62%, which shows that in Phenolphthaleate stenotropha, it is not an obligate sulfur. Some level of outcrossing do occur. And we also noted that African yam sets fruits and seeds when open pollinated, isolated, selfed, or crossed with other accessions. We also looked at the DNA purity level determination of African yam bean. Initially, we tried to use cowpea as a reference standard, but it gave us a wrong result. It gave us diploid. We have to use a reference standard of the same species. Luckily, Adesoye et al. Worked on African yam bean TSS5, which is one of the accessions I studied, and it had shown that it had a diploid number of 22. So TSS5 was taken as the reference standard, and the channel number was set at 100. So when we ran the flow cytometry analysis for the other 39 accessions, they all had their peak at the same channel that it is diploid. Flow cytometry can also be used if you carry out this technical flow cytometry in the ploidy level of the crop. So flow cytometry is faster and gives more accurate ploidy examination than a root tip chromosome counting, which is tedious. Definitely you need experienced histologists. And the chromosome of African yam bean is small. Sometimes there might be mistakes in counting. That's why for ploidy analysis, flow cytometry will be useful. I also looked at genetic variation in the proximate and phytochemical components of African yam bean, and found out that protein and carbohydrate are the 2 major components. We had an average protein content of 22.38% and carbohydrate of 57.17%. Accession TSS51 from IIT had the highest. We also looked at the tubers, because I noticed that in most of the literature, work has been done mostly on seeds producing tubers. The 4 tubers that I showed, 4 accessions, were also used for this analysis. I found that we had an average of 15.9%. The highest tuber had the highest, the accession that had the highest protein content in the tuber was TSS 107 from IIT, with a 15.9. Auto tubers are rich in protein. Unfortunately, in West Africa and mostly in Nigeria, the tubers are discarded, probably because of the fear that they may be poisonous. But in East and Central Africa, they consume the tubers in the tuber and decided to carry out this analysis on the tubers, which shows that it has substantial amount of protein. Also, the seeds were rich in magnesium, phosphorus, and potassium, while magnesium and potassium were prominent in the tubers. We decided to look at the anti-nutritional and nutritional content. Here, what I did was to compare the seeds and the tubers, and you see that the tubers had very low concentration of anti-nutrients, especially TSS 140 and 107, which had little or no nutritional content. This prompted me to go further to look at the effects of African yam bean seed and tuber flour mills on. We fed them tuber. We acclimatized the stew, which is their normal food. And we had 4 other groups fed with 50% tuber flour meal, 50% seed, and 100 to have more effect on the rats. For example, it had higher levels of and alanine aminotransferase, to cell effect on the liver. Oxidative, that the rats consumed less of that of the seed. And when you look at the additional comparison we saw, you know that the seed has. Finally, we look at the in vitro studies of African yam bean, because we say that we need to improve on African yam bean if in future we have to use biotechnological to have an efficient protocol for African yam bean. So what we did was to look at the use of X and other X plants and study morphogenic response of two African 5 and TSS 154. And we noticed that it was genotype dependent. Roots and root length in TSS 1 milligram per liter BAP for kinetic roots directly, and we acclimatized 75% of their session, peat pellet and vermiculate. MSc student is working on somatic embryogenesis of African yam bean, which will be useful for virus-free plants. Thank you for your attention.
Thank you, Dr. Bernard. It's impossible to have your presentation of 6 years in 5 minutes. We enjoyed the presentation, and we know that we are going higher. By my time here, we are due for lunch. So we'll be going now for lunch break. And when we come back, we shall converge at 1:30. Then we shall have Dr. Bamidele Michael, as he will be presenting in vitro studies of African yam bean, Sphaerostylis tendocarpa. So please enjoy your lunch. Emily, I think you will lead us to the kitchen. Thank you.